So today I'm going to be talking about stem cells. Stem cells naturally reside in the human body. They self-renew and proliferate without limit, and then they become specialized cells. They replace damaged cells and restore tissue and promote function in damaged tissues. And so in this way they act as an internal repair system. So embryonic stem cells, first an embryo is fertilized in vitro, and then they develop into totipotent cells, which differentiate into different cell types. And then they form blastocytes, which are cells that are formed in early development of mammals. And then they're cultured, creating millions. If the cells are allowed to clump, they form embryoid bodies that spontaneously differentiate. Scientists can control the characteristics of the stem cells by changing the medium of the chemical composition of the culture medium. And then they can also insert specific genes into the cells. Mesenchymal cells are collected from amniotic fluid during amniocentesis. Amniocentesis is a procedure that is done in pregnant women where they remove amniotic fluid to treat it or test it. Adult stem cells are gathered from bone marrow, blood, or the brain, and they're undifferentiated, so they are found in many tissues such as like neurons, heart tissues, blood cells, brains, tissues, liver tissues, muscle tissues, skin tissues. And they can differentiate into different cells among tissues, and that's known as transdifferentiation. Stem cell therapy, there's a lot of research on how to control the differentiation of stem cells to control birth defects and cancer. Pluripotent cells allow the testing of drugs, so they can see if drugs influence what they differentiate into. And cell-based therapies are renewable, unlike transplants, so they're much better. They can be used to treat a bunch of diseases such as macular degeneration, spinal cord injury, strokes, burns, heart disease, diabetes, osteoarthritis, Alzheimer's disease, autism, and rheumatoid arthritis. So stem cell transplants, there's two different types. The first is autologous, which uses a patient's treated stem cells. So they remove the stem cells, treat the stem cells, and reinsert them. Allogenic uses the donor stem cells. So they also have to undergo several intravenous infusions. They have to have stem cells, red blood cells and platelets, drugs, antibiotics, fluid, and chemotherapy. So the chemotherapy is to make room for the new stem cells in the bone marrow. And since they have to have so many intravenous infusions, they have to use a central venous catheter so that they don't do damage to the veins. So there are two cases of previous patients with stem cells that hold hope that stem cells could be a treatment for HIV. So the first case was a male patient from the UK. He received a stem cell treatment, and it was for a case of Hodgkin's lymphoma that he was diagnosed with in 2012. But he also had a previous diagnosis of HIV in 2003. And so after the transplant, he no longer tested positive for HIV, and they think this could be because the donor carried a genetic mutation that prevents the expression of an HIV receptor. There was also another case in Berlin, and the patient was being treated for leukemia. He also had a previous case of HIV, and he became HIV negative after the stem cell treatment.